Today we're going to discuss supervised learning in recurrent neural networks. By the end of this video, you'll be able to understand backpropagation through time, including its strengths and limitations in terms of both performance and biological realism. You'll be able to build and write out the equations for distinct RNN architectures and talk about how they deal with common issues to training RNNs over long timescales. You'll also be able to describe the principal ideas behind a couple of biologically inspired learning rules for RNNs, and describe the potential benefits of and best practices for applying RNNs to understand the neural mechanisms of cognition. So in a previous video, we went over the basic or vanilla RNN. So an RNN, if we unroll it in time, can be thought as a special case of a deep feed-forward neural network, where each time point in the RNN corresponds to a layer in a feed-forward neural network. However, there are a couple of twists to this story that, because of the recurrent connection being used at each time point, the same weights and biases are reused over and over again in recurrent neural networks, and that's not necessarily the case in feed-forward neural networks. Also, in contrast to most feed-forward neural networks, you can output information at each point in time, and you can also input information at each point in time, whereas in most feed-forward neural networks the inputs are just at the first layer and the outputs at the last layer. Here we're briefly going to mention another type of commonly used loss function. So far we've only seen the squared error. The squared error is commonly used for regression tasks where you have to predict a particular value, but the cross-entropy loss is often used for object recognition or classification tasks. And the reason this works well is that if there is a particular object in an image and you detect it, the cross-entropy is very low, whereas if it's not there, the loss grows very high, so it penalizes this quite strongly, and much more strongly than the squared error that we saw before. So this is not something particular to RNNs, but I thought we should mention another loss exists at least. Imagine that we're now doing a classification task with an RNN and we have this particular loss function. Well, we are able to output at each time point whether the network thinks this particular object is there, and at each time point we can compare the prediction to the true labels, and this allows us to generate a mini loss at each time point, and all of these mini losses are then summed up to the total loss for this particular training example. So the backpropagation in an RNN is just a particular application of the general backpropagation algorithm to an unrolled RNN. So we just start from the loss and we work backwards through the network, and in particular we work backwards starting from the loss at the final time point, and we move all the way back and propagate this error through time to the earlier parts of the network. And because we use these same weights at each time point, the weight updates for each time step get accumulated to a total update to particular weights. So the key thing to understand here that is different from most feed-forward neural networks is that we repeatedly multiply in the backpropagation by the same term, which is the transpose of the weight matrix used to propagate activity forward in time. This might be a little bit easier to understand if we replace a weight matrix by a single weight value. In this case, imagine our weight is equal to 2, and in order to do backpropagation from the final time point t all the way back to time point 1, we need to multiply the other terms by 2 raised to the number of time points. So imagine that we had 100 time points or time steps with this weight 2, then we would be multiplying all of the other terms by 2 to 100 when we're calculating the gradient. So our estimate of the gradient at the first time step is going to be multiplied by this incredibly huge number. And this is known as the exploding gradient problem. So if we imagine that this is how the loss function changes with respect to parameter values, This could be, for example, our particular weight here. With this exploding gradient problem, although we might know the right direction where the gradient decreases or increases locally, we're going to estimate that we should make a huge step in the parameter values in order to decrease the loss. However, this is going to make us very likely to overshoot and completely miss local minima of the loss function, and therefore to perform the task badly. In contrast, if we look at a weight value that's smaller than 1, for example 0.5, likewise for 100 time steps, we're going to multiply any other terms by an extremely small number. It's going to make it extremely difficult to learn or update our parameters at all based on activity many time steps ago. And this is called the vanishing gradient problem, whereas for the values greater than 1, it's called the exploding gradient problem. Now in practice, instead of dealing with single scalar weights, we're usually dealing with weight matrices, and we come up against the exploding gradient problem if the largest eigenvalue of the recurrent weight matrix is greater than 1, and if the largest eigenvalue of the recurrent weight matrix is smaller than 1, that is when we are vulnerable to the vanishing gradient problem. Now, people in the machine learning community have come up with a bit of a hack to mitigate the exploding gradient problem, and this is called gradient clipping. Now, because with the exploding gradient problem, you still have a good estimate of the correct direction of how the loss will change with an increase or a decrease in your parameter value, but you just have a poor estimate of the amount that you should shift the parameters. So what the proponents of gradient clipping suggest is that when regular gradient descent proposes a very large step, very large change to the parameters, we then reduce the step size to make it less likely to overshoot this local minima. And we can see here an example. If we have an accurate estimate of the direction of the gradient and we reduce the step size, well, we may be more likely to fix this problem. And the vanishing gradient problem for vanilla RNNs means that the algorithm believes essentially that activity in early time points will not affect the loss, but this is often not the case at all. But this isn't because early time points don't actually influence the loss. It's just because the backpropagation through time process struggles to recognize the influence of activity at an early time point on the loss, and it struggles to act upon their influence. In a previous video, we've seen the leaky RNN, which is a version of the continuous time RNN that has been discretized and is commonly used even traditionally in computational neuroscience. And for the leaky RNN, as a quick reminder, we start with the hidden layer activation value of the previous time point, and we update this in proportional to the time step divided by the time scale of individual units, and there's a leak term that pushes the activity back down towards 0 unless there are some synaptic inputs from external sources and the recurrent weights and a bias. In computational neuroscience, we often additionally add a noise term that we don't need to worry about here. To ease comparison with some models that we're going to see in a few moments, let's simplify this a little bit and consider delta t over tau as beta. So that our equation becomes this if we just neglect the noise term for simplicity. And we can rewrite this so that the activity at time point t is equal to 1 minus beta times the activity at time point t minus 1 plus beta times the new inputs. The new activity is equal to 1 minus beta times the old activity plus beta times the new inputs. So 1 minus beta is how much of the old stuff you remember at the new time point, and beta is how much you update. Now you notice that if we set beta equal to 1, we just get back the vanilla RNN, and our new activity is completely dictated by the external and recurrent inputs at the current time. Now, what does backpropagation through time look like for a leaky RNN? Imagine that we wanted to backpropagate the error to activity at time t minus k in order to see how much of this error is due to activity k time steps back. In order to do this, we will again use our chain rule for calculus, and we'll go back from the loss step by step with many terms like this. Now we can see that this, if we take the partial derivative of at with respect to at minus 1, we're going to get 1 minus beta here. And on the right hand side, we're going to get the derivative of our activation function times the recurrent weights times beta. We're going to see the same term again when we move one step further back in time, and we're going to have to multiply these things together many times. So the terms 1 minus beta and the recurrent weights are multiplied over and over again as we backpropagate through time. Therefore, if on each backpropagation step, if we multiply by a number that's close to 1, the gradient vanishes slowly. This will allow us to use activity from a bit further in the past to influence training, but it depends on this beta term, which in practice depends on our value of the neuronal timescale tau in comparison to the time step delta t. So now we're going to move on to some related models from AI. And each of these models uses at some point the sigmoid activation function, which we've mentioned briefly in past videos. But here we're just going to develop our understanding a little further. So the sigmoid activation function is just e to the x over e to the x plus 1. And what this does is it squishes any input on the x-axis here into the 0 to 1 range on the y-axis. And it has these smoothly varying properties which makes it differentiable. Because it squishes any input to the 0 to 1 range, it may be appropriate for tasks that require you to calculate the fraction or probability of something. So the first new model that we're going to encounter in this video is the light GRU, or light gated recurrent units. So we just went over the leaky RNN equation, and here we saw how a scalar value beta essentially gates or decides how much of the old information is retained and how much new inputs update the value of the activity. And this is just one number, a scalar value beta, which controls the time scale of remembering or updating for all units. A light GRU uses essentially the same equation, but now beta is a vector such that each element of beta controls the gating or updating of information for each recurrent unit in the RNN. And so a different number controls the timescale for each unit, and this can change in time because the gate beta is in fact learned, and it depends on both the external inputs and the recurrent activity. So we see that the gate has an equation that is like a vanilla RNN equation, but the activation function used here is usually the sigmoid. And this gate controls the fraction of newly calculated activity that will contribute to the next activity state, AT, of each unit. And in this paper by Ravanelli and Joshua Bengio and colleagues, they proposed using the ReLU activation function for the other units. And they also proposed another additional detail, which is to normalize the activations across units within a layer. By normalizing, they just subtract the mean and divide by the standard deviation plus a small number. And this is quite similar, at least superficially, to something that happens in local neural populations in the brain. And in the brain, this idea is called the normalization canonical computation. And it's called a canonical computation because it seems to happen in many different parts of the brain very commonly. So another activation function that is used in similar networks is the tanh, and this is very similar to the sigmoid, except instead of squishing all inputs to the 0-1 range, it squishes inputs to the -1 to 1 range. So although we're building up a layer of complexity of the model, we're actually stepping backwards in time. So the light GRU came as a simplification of the GRU, or gated recurrent units. And we can see that the GRU equation here looks exactly the same as the light GRU equation, but it additionally has another gate, and this is a separate gate on previous time step activations that occurs at this linear stage of the update equation. So the GRU has two gates, and these two gates, beta and gamma, are learned, and again they depend on the inputs and the recurrent activity at a particular time point. So similar to the light GRU, gives you the possibility of dynamic gates that are particular to individual recurrent units in the RNN. So we see in the original GRU they used a tan-H activation function, and this differed in the light GRU update. So let's talk a little bit about why they proposed a couple of those updates. Well, one, generally in 2018 it became obvious from the feed-forward deep neural network literature that in many cases ReLU activation function was more effective for training very deep networks. Part of the reason is because for the sigmoid and tan-H, if you get very high or very low inputs, the gradient becomes very close to zero, which makes it very difficult to learn. Whereas for the ReLU, at least for high values, that's not the case. And one difficulty, which meant it wasn't used earlier in recurrent neural networks, was that you faced the issue of potentially exploding gradients and extremely high activities. However, they attempted to counteract this in the light GRU paper by introducing the batch normalization, which we just mentioned. They also proposed removing the second gate because they found, at least in some of the tasks that they studied, that the activity in the two gates in the GRU was quite strongly coupled, and perhaps they were not both needed. However, I think it's still an open question as to which of these models might be more helpful for us in terms of trying to understand recurrent computations in the brain. So the GRU is itself a simplification of a model that is proposed way back in deep learning prehistory, 1997. So the LSTM, or long short-term memory network, has been extremely successful at solving these vanishing gradient problems and has excelled at a range of tasks that require temporal dependencies, including machine translation. So LSTMs maintain two separate activity variables that they sometimes call long-term and short-term memory variables. And they control how these long and short-term memories are updated by the use of three different gates. And each of these gates has the same form. It's a sigmoid activation function of learned external inputs and recurrent activity and a bias at the current time. However, each of these gates learns its weights and biases separately, so in practice they work independently. And each of these gates controls the fraction of something contributing to a new memory state, and we can interpret it as a fraction again because they use the sigmoid activation function. Now, where the LSTM differs from most other RNNs is in the use of this internal cell state that we can think about as a slightly longer term memory. And the update for the long-term memory is as follows. The new long-term memory is the fraction that you don't forget of the old long-term memory, plus the fraction you allow in of the candidate new long-term memory. This is quite similar to the update rule that we've seen for the GRU and light GRU, and even the leaky RNN. However, in all of those cases, we just used one variable beta, which decided how much you update your memory, and it was the 1 minus that beta that decided how much you hold on to the old memory. However, in the LSTM, these two gates are independent, so that you can hold on to the entirety of the old memory and add on some fraction of the new information. And this new candidate long-term memory is itself a tan-H of the inputs at the current time point from recurrent and external sources. So the equation to propose the candidate new memory is exactly the same as for the gates, except it uses a tan-H activation function instead of a sigmoid. Similarly, the output to the next time point, or the new short-term memory activity, is just the fraction that you let out from the third gate of a new candidate short-term memory. And that candidate short-term memory is just the long-term memory put through a tan-H activation function. So again, the amount that the longer term memory state contributes to the new shorter term memory or activity state and how that affects the next time point is controlled by this output gate O. However, it's important to note here that the long-term memory in this LSTM model doesn't really seem to be the same thing as long-term memory in the brain. In particular, this is something about some persistent activity state that can stay over many time steps, whereas in long-term memory in psychology and neuroscience, this is something that can go away completely. You're not constantly thinking about it. It doesn't seem to be constantly held in neural activity, and it can be recalled sometimes many years later. So I think although it may be helpful to separate this more persistent memory state, CT, from a more rapidly changing memory state, HT, it maybe is a bit unfortunate to call it long-term memory. So now we're going to speak a little bit more about how LSTMs deal with the vanishing gradient problem. So the computation graph for LSTMs is quite a bit more complicated than what we've seen previously for simple feed-forward and recurrent neural networks. So to calculate how the loss depends on activity several time steps ago, we can propagate back the error through many different paths. But in order to avoid vanishing gradients, we just need at least one of these paths to not vanish. And this will ensure a steady flow of the gradient back in time. Here we can see that there's one very direct path by which we can propagate the error back in time that was explicitly designed to reduce this vanishing gradient problem. Here we can see how the loss depends on the long-term memory activity, k time steps ago. And we can see that there are a lot of these recursive looking computations like we saw in the RNN. But here it's depending on this long-term memory state. So here we remind ourselves of the equation for the long-term memory activity, and the partial derivative of this with respect to ct minus 1 on the left-hand side is just f of t, or the value of this gate f, which is also known as the forget gate. And on the right-hand side, well, this could depend also on ct minus 1, and that could perhaps vanish. But we only need one part of the gradient to not vanish to propagate the gradient back in time. So what that means is if the forget gate several time steps ago had very low values, then we may indeed see a vanishing gradient. But that's actually okay, because if the forget gate was very low, then activity from before then will not affect activity at the time point we're currently assessing. So therefore the forget gates regulate the gradient flow according to how much a long-term memory in state ct minus 1 contributed to the next state ct. So there's very nice in-depth explanation of this by Mitesh Kapra on YouTube, and I've put a link to one of them in the references at the end of this video. So we've now seen a number of these developments in RNN architectures that come from the AI literature, and they essentially put in different numbers of gates within the system that allow errors to be propagated further back in time in order to increase the amount of learning that can be done from distant time points. However, what about the biological realism? We really care about what's going on in the brain. And leaky RNNs are very close to traditional untrained recurrent neural network models used in computational neuroscience. And in fact, they remain quite popular in applying AI to neuroscience, as leaky RNNs are often observed to have brain-like dynamics in their neuron activities. This isn't always true for other RNN architectures, including the vanilla RNN, the various versions of the GRUs and LSTMs. However, other models are more effective at learning long-term dependencies, and some efforts have been made over the last few years to develop more biologically plausible versions of these more powerful GRU and LSTM architectures. Here we can see some proposals for a biologically inspired variant of the LSTM by Rui Costa and colleagues. And one of the key differences here is that instead of this multiplicative gate that is used in the regular LSTM, whereby the value of the gate between 0 and 1 is multiplied by the activity value in order to reduce it multiplicatively, as shown here, we see subtractive gating, which is thought to be representative of how inhibition from at least some inhibitory neurons acts on the excitation of excitatory cells in the brain. And there have also been biologically inspired variants proposed of some of the other architectures, including the light GRU. So please turn to the blackboard and answer the first quiz question. We talked previously about the biological implausibility of the backpropagation algorithm when considering deep or feed-forward neural networks. And what about the backpropagation through time? So similarly to regular backpropagation in feed-forward neural networks, backpropagation through time requires a reusing of the same weights that are used to propagate activity forward in time in order to assign the credit or blame for error to particular activities and weights at previous time points. But also uniquely to backpropagation through time and recurrent neural networks, for this to work, it requires the same neuron to be able to store and retrieve with perfect accuracy the values of its activities from all points in the past. And at least the strict version of this seems to be something that neurons in the brain do not do. So there have been some suggestions for more biologically inspired or biologically plausible learning in recurrent neural networks. Here's one proposition from Murray. And in this biologically inspired learning rule, the change in weight between neurons A and B at a particular time t depends on a learning rate and the error, which in this case is distributed in proportion to random weights, and a moving average of recent co-activations between neurons A and B. So this proposition has many strengths. It only depends on information that each neuron should actually have. So this doesn't suffer from the same problem of biological implausibility as backpropagation, where updates to weights require information of weights and activities of neurons many synapses away. There's also no need to reuse the weights in the forward and backward directions. And here he uses the same idea from the paper of Lillicrap and colleagues that we studied in the Supervised Learning in Deep Neural Networks lecture. And they find that it works similarly well in the recurrent case. Essentially, the feed-forward weights learn to align with these random feedback weights and approximate the full backpropagation algorithm. There's also no need in this learning rule to rerun the activations backwards at the end of the trial in order to learn. All of the learning takes place online in a step-by-step manner. One limitation of this is that the co-activations are actually forgotten at a rate determined by the neuronal timescale. And there's a paper by Owen Marshall and colleagues that showed that this is effectively similar to backpropagating about one time step, so perhaps not as powerful as the full backpropagation through time algorithm. There is another perspective on the need to run activations backwards at the end of the trial to learn. In fact, it turns out that the brain, or at least parts of the brain, including the hippocampus, seem to do something similar to this. Backwards replay of activity does happen in the brain, and it does seem to happen quite often at the end of trials, perhaps when the animal is learning from its experiences. So Cheng and Brown proposed a biologically inspired learning neural network that they described in their preprint on bioRxiv. And there are many details here, but I think the interesting one for us is that they have proposed to connect one RNN to a second RNN that looks a lot like a Hopfield network that we studied several videos ago, but with one crucial difference. So in this variant of a Hopfield network, the weights between neurons activated by consecutive inputs are strengthened. So there are actually stronger connections between neurons activated by consecutive inputs. And this allows the network to run back and play through a sequence of past activations, and also to play that backwards, which looks quite similar to what you might want to do if you are running something like backpropagation through time in order to learn. So I think this biological plausibility of backpropagation through time is still somewhat of a hot topic, and perhaps there are some variants or some changes that can be made so that the brain could do something similar to backpropagation through time, or perhaps it uses a completely different learning algorithm. But perhaps even if this learning algorithm is not biologically plausible, networks learned with backpropagation through time could perhaps still tell us something important about the brain. And now we're going to see a couple of examples of supervised learning applied to RNNs in the context of neuroscience. And these networks were both trained with backpropagation through time. And this is really a landmark paper in the use of trained recurrent neural networks in neuroscience. And they were using this approach in combination with electrophysiological recordings from the monkey cortex to try and understand how the brain uses context to guide behavior. So Mante, Cicillo, and Newsom trained monkeys to perform the following cognitive task. Monkeys were trained to keep their eyes on a particular spot marked by a particular shape. This is known as the fixation cross. And the particular color or shape of this fixation cross provided the context cue for the monkey. After about 300 milliseconds, two targets were displayed on the left and right side of the screen, and these were colored green and red. Then for 750 milliseconds, or 3/4 of a second, the monkeys were shown moving dots, similar to what we saw previously, but now the moving dots are not just moving in a particular direction plus noise, but they also have a particular color, such that on any individual trial, dots could be moving predominantly to the left or to the right, and there could also be a majority of green or red colored dots. The dots are then removed and there's a short delay, and whenever this fixation cross disappears, you have to make a saccade. And the rule is as follows. For a particular context, for example, a blue cross, this will mean that the monkeys should pay attention to the motion direction but not the color, so that if the motion direction is to the right, they should, at the end of the trial, move their eyes to this spot on the right hand side of the screen. However, if the context cue is different, for example, a yellow cross, then they should pay attention to the color, and they should, at the end of the trial, move their eyes to the color of the spot corresponding to the majority of the dots. So for example here, to me there seem to be more green than red dots, so that at the end of the trial, in the color context, I should move my eyes to the left. Now here we can see an example of many different stimuli with increased motion coherence, so most of the dots moving to the left over here, and most to the right in these columns, and very little difference between the left and the right in the middle. And we can also see that there is increasing or decreasing amounts of color coherence, with most of the dots being red or green or a real mix in the middle. And this dictates some of the difficulty in solving the task correctly. And here we can see that the monkeys learned to perform this task really quite well. On the top row, we can see the results for the motion context. So this is when monkeys were meant to respond with an eye response according to the predominant direction of motion of the dot. And you can see that the monkeys do this really well, so that when most of the dots are moving to the right, they respond quite successfully by saccading their eyes to the right hand side most of the time, and vice versa for the left. However, when the color coherence changes, this only causes a very slight bias in their behavior. Likewise, in the color context, when they're meant to ignore motion and focus on the color, they tend to do this very well. So it seems that monkeys are able to use context to guide their behavior in this perceptual decision-making task quite well. And one thing to point out here is that we can compare trials where the sensory stimulus is exactly the same, but it's just the contextual clue that differs between the trials. And this leads to a robust difference in behavior for the monkeys according to a different context for the exact same stimulus. So how does this actually occur in the brain, that a particular sensory stimulus can lead to two different behaviors based on the context? And they were interested in looking at how the neural dynamics may change according to the context. And in order to do this, they look at something called the state space of neural activity. And we can imagine this as follows. Say we're recording from just 3 neurons, and if neuron 1 is highly active at the start of the trial, and neuron 2 and neuron 3 have quite low activity, then we could plot the activity in this state as corresponding to high on this axis and low on these other axes. So then if we track the activity of these 3 neurons in time, this will trace out a trajectory in this neural state space until we reach a final point in the trial. So in reality, neuroscience experiments record from many more neurons. So we take the same idea, but this space is much higher dimensional, corresponding to the higher number of neurons. And this can make it difficult to interpret. So commonly, neuroscientists will use dimensionality reduction techniques, where principal components analysis, or PCA, would be one example of that. So the authors of this paper used a variant of PCA that allows you to assign these orthogonal dimensions of neural activity to particular aspects of the task. In particular, they were able to isolate a direction corresponding to neural activity that responded to motion, one that responded to changes in color, and a third that was related to the final choice of the monkey. And at the beginning of the trial, when there was no stimulus, this activity started in the center of this space, about 0 on each axis, and then it proceeded on a trajectory until a choice was made. So here we can see some of the real neural activity data from their experiment, and here we can see it plotted on the motion and choice axes. And what we can see here is the activity for motion stimuli of different strengths corresponding to one direction and another. And we can see here that strong motion stimuli in this particular direction lead to this big increase in activity along the motion axis, which gradually leads towards a positive choice in this direction. In contrast, a strong motion stimulus in the other direction leads to a decrease in this motion axis, followed by a choice in the other direction. If we then look for these same stimuli, not at activity between these choice and motion axes, but at activity defined by these choice and color axes, we see that on the color axis there is very little difference in neural activity throughout the trial. However, if we now group the stimuli according to their color, which was actually irrelevant to the task in this context, we see that this neural activity strongly reflects the differences in color of the different stimuli, even though it was irrelevant to the behavior. And we can imagine this, we can understand this as follows, that differences in the color are strongly represented along this color axis in neural activity, but they ultimately do not affect the choice, with the same choice being made regardless of the color and solely depending on the motion direction. And we see equivalent results in the opposite direction for the color context. So this suggests that color information in the motion context indeed reaches the prefrontal cortex, this area, the frontal eye fields, but it does not affect choice. And this is represented in these schematic diagrams of their results, where motion direction in one direction, in this case the vertical up direction, corresponds to a left choice, and motion input in the down direction corresponds to the opposite choice. However, extremes of green or red can both correspond to the same choice in the motion context. This was actually very surprising, because the dominant hypothesis for how the brain would solve this context-dependent sensory choice was through a mechanism called early selection, whereby sensory information is gated out before reaching the prefrontal cortex. And this is what it would look like. In the color context, you would not be able to detect any motion signals in the prefrontal cortex. And in the motion context, you would see motion input aligned directly with this choice axis. And they also ruled out many other leading alternative models. And this led them to a situation in which none of the previously proposed neural models of context-dependent decision-making were sufficient in order to describe their neural data. So this is when they turned to training recurrent neural networks. and they analysed how a leaky RNN could use context to guide behaviour. And let's go through a little bit the steps of how they do this, because I think this is really great practice for applying RNNs and trained neural networks to neuroscience data. So on the left, we're going to show the general steps for the method, and on the right, we're going to show how it was implemented and results relevant to this paper. So the first step is to train the recurrent neural network on the same task as the individual, which could be a human, a mouse, or as in this paper, a monkey. And in this paper, they trained a leaky RNN with backpropagation through time on the same context-dependent decision-making task. The next step is to analyze the RNN using the same methods used to analyze the real neural activity. And they found that the RNN activity resembled the real neural population activity seen in the prefrontal cortex much better than any of the previously proposed hypotheses or alternative models. And the third step is, if this second step has shown that the RNN is a good match to the neural data, then you can perform additional experiments and analyses on the RNN that may be infeasible or very costly to perform on an animal or human subject. And these analyses on the RNN will hopefully enable deeper insights into potential neural mechanisms and perhaps suggest tests for future experiments. So in this paper, they did a dynamical systems analysis on the RNN activity. So without getting deep into the dynamical systems theory, they found that the context signal modifies the dynamical landscape, which affects how the neural responses to motion and color guide activity towards making a choice along this choice axis in neural activity space. So this is really a remarkable achievement in applying RNNs and dynamical systems analysis to understand neural activity in the brain. And this kind of careful application of RNNs can be really useful for generating new hypotheses about how the brain performs its functions. So another reason to think about using supervised learning and RNNs is to develop computation models of more complex cognitive tasks and functions, and things that have not been yet successfully tackled using hand-built or hand-designed neural networks. So the traditional paradigm in computational neuroscience has been to design a particular network to solve a particular task. And even when AI type models are applied in a neuroscience context, usually start from a blank slate, completely untrained RNN, and train it to solve one particular task and then analyze its activity. However, the real brain, when it learns a new task, is not starting from a completely blank slate. It already knows many other things. So Guanyu Robert Yang and colleagues trained a single RNN, a leaky RNN, and in other cases a GRU, to perform 20 cognitive tasks from the neuroscience literature. And they did this with an interleaved training protocol that meant that as the network learned a new task, it would not forget other tasks that it was learning in parallel. And by using a neural state space analysis, similar to the ones we just saw previously, and analyzing neural activity across many of these tasks, they found that groups of neurons activated not for individual tasks, but for individual cognitive functions, such as neurons that responded to all of the tasks that required working memory, other neurons responded to all of the tasks that required decision making, and when certain tasks required combinations of these functions, we used a compositional solution in order to use combinations of these clusters of neurons that contribute to these different functions. So this provides at least a proof of principle that a brain or neural network could use a compositional solution in order to perform many different tasks with the same set of neurons. So more generally, training neural networks can teach us how to build computational models for aspects of cognition that were previously overly complicated for hand-designed networks. So please go to the blackboard and take the final quiz question for this video. So to recap, backpropagation through time is effective at assigning credit and blame to synapses, but it's not generally thought to be biologically realistic. And it also suffers from vanishing and exploding gradient problem, in particular for the basic or vanilla versions of the recurrent neural networks. And this prevents learning from time points in the distant past. So, however, several different architectural variants on the RNN have been proposed, such as the light GRU, GRU, and LSTM, to tackle the vanishing gradient problem. But their biological relevance is still debated, and variants on these architectures with more biological realism are being proposed frequently now. We also saw that some more biologically realistic learning rules have been proposed, and these take different approaches, ranging from completely avoiding storing history of past activity states to retrieving past activity states through more biologically inspired mechanisms, such as mechanisms inspired by hippocampal replay. We also saw that even if the learning rule used to train a network is not realistic, we can still learn some valuable information from the learned network. Sometimes these networks trained with backpropagation through time can give valuable insights and new hypotheses as to how the brain performs cognitive tasks or indeed other functions. So if you want to learn more, here are several papers and a YouTube video that goes into more detail of the topics we discussed today. Thank you.